everyone, welcome to a new session of the paper Cell Biology. In this session, we would study about the chromosome morphology, structure with an emphasis on the various levels of chromosome organization. We would also study about the histone and non-histone proteins, differentiate between euchromatin and heterochromatin. Chromosome structure. In the nucleus of each cell, the DNA molecule is packaged into thread-like structures called chromosomes. Each chromosome is made up of DNA tightly coiled many times around proteins called histones that support its structure. Chromosomes are not visible in the cell's nucleus, not even under a microscope when the cell is not dividing. However, the DNA that makes up chromosomes becomes more tightly packed during cell division and is then visible under a microscope. Most of what researchers know about chromosomes was learned by observing chromosomes during cell division. Each chromosome has a constriction point called the centromere, which divides the chromosome into two sections or arms. The short arm of the chromosome is labeled the P arm, the long arm of the chromosome is labeled the Q arm. The location of the centromere on each chromosome gives the chromosome its characteristic shape and can be used to help describe the location of specific genes. Not only are the genomes of most eukaryotes much more complex than those of prokaryotes, but the DNA of eukaryote cells is also organized differently from that of prokaryotic cells. The genomes of prokaryotes are contained in single chromosomes, which are usually circular DNA molecules. In contrast, the genomes of eukaryotes are composed of multiple chromosomes, each containing a linear molecule of DNA. The DNA of eukaryotic cells is tightly bound to small basic proteins or histones that package the DNA in an orderly way in the cell nucleus. Nucleosomes. Nucleosomes are the fundamental units of chromatin. They are found in all eukaryotes with the exception of dinoflagellates and are conserved in structure. The relationship between DNA and nucleosome can be investigated using nucleases. Limiting digestion of decondensed chromatin separates the nucleosomes into short fragments consisting of single nucleosome units. The DNA that remains is protected by the proteins of the nucleosome particle, which at this stage is termed a chromatosome. Further nuclease digestion reduces the DNA to 146 base pairs in length and liberates some protein from the nucleosome. This length of the DNA is conserved throughout the eukaryotes and is relatively resistant to further nuclease attack. It represents the DNA intimately associated with the proteins which comprises the nucleosome core. The particle thus formed, the nucleosome core particle, consists of a histone octamer containing two copies each of histones H2A, H2B, H3 and H4 and 146 base pairs of core DNA which is wrapped around this octamer twice. The core particles are joined together by linker DNA whose length varies between species and between different cell types. Histones. Histones are highly basic proteins rich in positively charged lysine and arginine residues which fold to form a compact core with a protruding N-terminal tail. The positive residues interact with DNA by forming salt bridges with the negatively charged DNA backbone, while the tails are targets for post-translational modification and facilitate nucleosome-nucleosome interactions and interactions with other chromatin proteins. In this table, you can see the various types of histones, the molecular number and the number of amino acid residues present in each of these histones. First one is H1. The molecular weight is 22,500. The number of amino acids is 244 and the percentage of lysine and arginine is 30.8. H2A, molecular weight is 13,960. The number of amino acids is 129 and the percentage of lysine and arginine is 20.2. H2B, the molecular weight is 13,774. The number of amino acids is 125 and the percentage of lysine and arginine is 22.4. H3, molecular weight is 15,273. The number of amino acids is 135 and the percentage of lysine and arginine residues is 22.9. H4, 11,236 is a molecular weight. The number of amino acids are 102 and the percentage of lysine and arginine is 24.5. 5 major families of histones exist: H1 or H5, H2A, H2B, H3 and H4. Histones H2A, H2B, H3 and H4 are known as the core histones, while histones H1 and H5 are known as the linker histones. 2 of each of the core histones assemble to form one octameric nucleosome core approximately 63 angstroms in diameter. 147 base pairs of DNA wrap around this core particle 1.65 times in a left-handed superhelical turn to give a particle of around 100 angstroms across. The linker histone H1 binds the nucleosome at the entry and exit sites of the DNA, thus locking the DNA into place and allowing the formation of higher order structure. All histones appear to undergo some form of post-translational modification, usually on the N-terminal tail, and in many cases specific patterns of modification correlate to changes in chromatin function. Histone tails are long and unstructured but ordered by nucleosome-nucleosome interactions. Non-histone proteins. The histones represent the bulk of protein in chromatin and are relatively homogeneous in nature. The remaining chromatin proteins, collectively described as non-histone proteins, represent a small but extremely heterogeneous fraction. The non-histone proteins include enzymes involved in DNA and histone metabolism, replication, recombination and transcriptional regulation. Roger Kornberg in 1974 described the basic structural unit of chromatin, which is called the nucleosome. The structural organization beginning from nucleosome to chromosome is explained in the following table. Here you find 3 levels of chromosome organization, which is represented schematically. The first level of packaging involves the winding of DNA around a protein core to produce a bead-like structure called a nucleosome. This gives a packing ratio of about 6. This structure is invariant in both the euchromatin and heterochromatin of all chromosomes. The protein core is composed of 8 histone proteins, 2 each of H2A, H2B, H3 and H4. Histone H1 forms the linker between 2 nucleosomes. 146 base pairs of DNA is wrapped around each nucleosome. The second level of organization includes the coiling of beads in a helical structure called the 30 nanometer fiber that is found in both interphase chromatin and mitotic chromosomes. This structure increases the packing ratio to about 40. This appears to be a solenoid structure with about 6 nucleosomes per turn. This gives a packing ratio of 40, which means that every 1 micrometer along the axis contains 40 micrometer of DNA. The stability of this structure requires the presence of the last member of the histone, gene family, histone H1. Because experiments that strip H1 from chromatin maintain the nucleosome but not the 30 nanometer structure, it was concluded that H1 is important for the stabilization of the 30 nanometer structure. The third level of chromosome organization, the fiber is organized in loops, scaffolds and domains that give a final packing ratio of about 1000 in interphase chromosomes and about 10,000 in mitotic chromosomes. The final level of packaging is characterized by the 700 nanometer structure seen in the metaphase chromosomes. The condensed piece of chromatin has a characteristic scaffolding structure that can be detected in metaphase chromosomes. This appears to be the result of extensive looping of the DNA in the chromosomes. Chromatin structure. The eukaryotic cell cycle produces remarkable changes in the structure of chromosomes. In non-dividing eukaryotic cells in the G0 phase and those in interphase such as G1, S and G2, the chromosome material, the chromatin, is amorphous and appears to be randomly dispersed in certain parts of the nucleus. In the S phase of interphase, the DNA in this amorphous state replicates, each chromosome producing two sister chromosomes called sister chromatids that remains associated with each other after replication is complete. Euchromatin. The lightly stained regions in chromosome when stained with basic dyes are called euchromatin and contain single copy of genetically active DNA. The extent of chromatin condensation varies during the life cycle of the cell and plays an important role in regulating gene expression. In the interphase of cell cycle, the chromatin are decondensed and known as euchromatin, leading to gene transcription and DNA replication. Heterochromatin. The word heterochromatin was coined by Emil Heitz based on cytological observations. They are highly condensed and ordered areas in nucleosomal arrays. About 10% of interphase chromatin is called heterochromatin and is in a very highly condensed state that resembles the chromatin of cells undergoing mitosis. They contain a high density of repetitive DNA found at centromeres and telomeres form heterochromatin. Heterochromatin are of two types, the constitutive and facultative heterochromatin. The regions that remain condensed throughout the cell cycle are called constitutive heterochromatin, whereas the regions where heterochromatin condensation state can change are known as facultative. Constitutive heterochromatin is found in the region that flanks the telomeres and centromeres of each chromosome and in the distal arm of the Y chromosome in mammals. Constitutive heterochromatin possess very few genes and they also lead to transcriptional inactivation of nearby genes. Chromatin structure is organized at several levels. The basic structure of chromatin, either heterochromatin or euchromatin, is called nucleosome. The nucleosome is a complex of 146 base pairs of DNA wound in two turns around the outside of a disc like complex of 8 proteins called histones. The histone core contains 2 copies each of 4 histones, H2A, H2B, H3 and H4. The histone octamer is wrapped by very close 2 turns of DNA. Linker DNA and another histone H1 join together the nucleosomes, which are about 65 base pairs worth. H1 binds cooperatively to nucleosomes so that a gene can be zipped up all at once by the binding of many H1 molecules successively. There are more than 1,000 different types of these proteins which are involved in a range of activities including DNA replication and gene expression. Histones are not found in eubacteria, for example E. coli, although the DNA of these bacteria is associated with other proteins that presumably function like histones to package DNA within the bacterial cell. The basic structural unit of chromatin, the nucleosome, was described by Roger Kornberg in 1974. Nucleosomes are packed into more compact structures. First, the nucleosome fiber is condensed in a helix-like array called the 30 nanometer fiber. Scaffolding proteins hold the chromatin domains together. For active genes, these domains form loops with the active sequences on the brushy part of the loops and the regions between genes attached to the scaffold. Non-histone proteins regulate the condensation and decondensation of chromatin. Again, active genes, those that are being transcribed, are present in the less condensed portions of the chromatin. Two types of experiments led to Kornberg's proposal of the nucleosome model. First, partial digestion of chromatin with micrococal nuclease was found to yield DNA fragments approximately 200 base pairs long. Packaging of DNA into such a 10 nanometer chromatin fiber shortens its length approximately 6-fold. The extent of chromatin condensation varies during the life cycle of the cell. In interphase cells, most of the chromatin, called euchromatin, is relatively decondensed and distributed throughout the nucleus. During this period of cell cycle, genes are transcribed and the DNA is replicated in preparation for cell division. Most of the euchromatin in interphase nuclei appears to be in the form of 30 nanometer fibers organized into large loops containing approximately 50 to 100 kb of DNA. In contrast to the euchromatin, about 10% of interphase chromatin, called heterochromatin, is in a very highly condensed state that resembles the chromatin of cells undergoing mitosis. Heterochromatin is transcriptionally inactive and contains highly repeated DNA sequences such as those present at centromeres and telomeres. As cells enter mitosis, their chromosomes become highly condensed so that they can be distributed to daughter cells. The loops of 30 nanometer chromatin fibers are thought to fold upon themselves further to form the compact metaphase chromosomes of mitotic cells, in which the DNA has been condensed nearly 10,000-fold. Such condensed chromatin can no longer be used as a template for RNA synthesis, so transcription ceases during mitosis. Centromere. The centromere is the part of a chromosome that links sister chromatids. During mitosis, spindle fibers attach to the centromere via the kinetochore. Centromeres were first defined as genetic loci that direct the behavior of chromosomes. Their physical role is to act as a site of assembly of the kinetochore, a highly complex multi-protein structure that is responsible for the actual events of chromosome segregation, that is, binding microtubules and cell signaling to the cell cycle machinery when all the chromosomes have adopted correct attachments to the spindle, so that it is safe for cell division to proceed to completion and for cells to enter anaphase. There are, broadly speaking, two types of centromeres. Point centromeres bind to specific proteins that recognize particular DNA sequences with high efficiency. Any piece of DNA with the point centromere DNA sequence on it will typically form a centromere if present in the appropriate species. The best characterized point centromeres are those of the budding yeast Saccharomyces cerevisiae. Regional centromeres is the term coined to describe most centromeres which typically form on regions of preferred DNA sequence, but which can form on other DNA sequences as well. The signal for formation of a regional centromere appears to be epigenetic. Most organisms ranging from fission yeast such as Schizosaccharomyces pombe to humans have regional centromeres. The centromeres thus serve as both sites of association of sister chromatids and as the attachment sites for microtubules of the mitotic spindle. They consist of specific DNA sequences to which a number of centromere associated proteins bind, forming a specialized structure called the kinetochore. The binding of microtubules to kinetochore proteins mediates the attachment of chromosomes to the mitotic spindle. Proteins associated with the kinetochore then act as molecular motors that drive the movement of chromosomes along the spindle fibers, segregating the chromosomes to daughter nuclei. Telomeres. Telomeres are the region of DNA at the end of the linear eukaryotic chromosomes that are required for the replication and stability of the chromosome. McClintock recognized their special features when she noticed that if two chromosomes were broken in a cell, the ends were sticky and end of one could attach to the other and vice versa. However, she never observed the attachment of the broken end to the end of an unbroken chromosome, suggesting that the end of chromosomes have unique features. Telomere sequences remain conserved throughout vertebrates and they form caps that protect the chromosomes from nucleases and other destabilizing influences, and they prevent the ends of chromosomes from fusing with one another. The telomeric DNA contains direct randomly repeated sequences of the form T or A X G Y, where X is between 1 and 4 and Y is greater than 1. Human telomeres contain the sequence TTA GGG repeated from about 500 to 5000 times. Certain bacteria possess telomeres in their linear genetic material, which are of two types. One of the types is called a hairpin telomere. As its name implies, the telomeres bend around from the end of one DNA strand to the end of the complementary strand. The other type of telomere is known as invertron telomere. This type acts to allow an overlap between the ends of the complementary DNA strands. Chromosome morphology. Chromosomes were first seen by Hofmeister in 1848 in the pollen mother cells of Triscantia in the form of darkly stained bodies. The term chromosome means chrome means color and soma means body was used by Waldeyer in 1888 to designate their great affinity to basic dyes. Chromosomes are the most significant components of the cell, particularly they are apparent during mitosis and meiosis. Their presence was demonstrated long before they were named chromosomes by Waldeyer in 1888. A chromosome can be considered as a nuclear component having special organization, individuality and function. It is capable of self-reproduction while maintaining its morphologic and physiologic properties through successive cell divisions. The morphology of chromosome can be best studied at the metaphase or anaphase of mitosis when they are present as definite organelles being most condensed or coiled. Number. The number of chromosomes in a given species is usually constant, containing diploid number, that is 2n, of chromosomes in their somatic cells and haploid number, that is n, of chromosomes in their sex cells, which are the sperms and ova. The number of chromosomes is variable from 1 to several hundred among different species. For example, in Ascaris megalocephala it is 2, while in certain protozoans such as Agreta there are more than 300 chromosomes, in Paramecium 30 to 40, in Radiolarians as many as 1600, in Hydra vulgaris 32, Musca domestica 12, Rana esculenta 26, Columba livia 80, Orictolagus uniculus 44, Gorilla 48 and Homo sapiens 46. The chromosome numbers are also helpful for taxonomy. In the angiosperms, the most frequent haploid number is 12 and members of this group have a range from 3 to 16. Similarly, in fungi, haploid number ranges from 3 to 8. In primates, this haploid number is from 16 to 30. This haploid set of chromosomes present in the nucleus of gametes, while in a diploid cell there will be 2 genomes. The diploid cells are the somatic cells in the body. The diploid cells gets the diploid set of chromosomes by the union of the haploid male and female gametes in the sexual reproduction. Size. Chromosomes range on an average from about 0.5 to about 30 microns in length and from 0.2 to 3 microns in diameter. The relative number of chromosomes generally differ in the nucleus, but at a time all chromosomes of a cell may be of the same size. Plant cells normally possess larger chromosomes than animal cells. Trillium has chromosomes which may reach up to the length of 32 microns at metaphase. Monocotyledon plants usually have larger chromosomes than the dicotyledons which contain greater number of chromosomes. Among the animals, grasshoppers, crickets, mantids, newts and salamanders have large chromosomes. Shape. The shape of the chromosomes is changeable from phase to phase in the continuous process of the cell growth and cell division. In the resting phase or interphase stage of the cell, the chromosomes occur in the form of thin coiled elastic and contractile thread-like stainable structures, the chromatin threads. In the metaphase and the anaphase, the chromosomes become thick and filamentous. Each chromosome contains a clear zone known as the centromere or kinetochore along their length. The centromere divides the chromosomes into 2 parts. Each part is called chromosome arm. The position of centromere varies from chromosome to chromosome and it provides different shapes to the latter, which are the following. 1. Telocentric. The rod-like chromosomes which have the centromere on the proximal end are known as the telocentric chromosomes. 2. Acrocentric. The acrocentric chromosomes are J-like in shape, but these have the centromere at one end and thus giving a very short arm and an exceptionally long arm. Submetacentric. The submetacentric chromosomes are L-shaped. In these, the centromere occurs near the center or at the medium portion of the chromosome and thus forming 2 unequal arms. Metacentric. The metacentric chromosomes are V-shaped and in these chromosomes the centromere occurs in the center and forming 2 equal arms. The amphibians have metacentric chromosomes. Let us now summarize what we have learnt in this session. Chromosomes are highly ordered structures. Eukaryotic DNA forms a complex with histones and other proteins and is packaged into chromosomes. Some of the DNA is permanently condensed into heterochromatin while the rest is condensed only during cell division. The basic structure of chromatin, either heterochromatin or euchromatin, is called the nucleosome. The nucleosome is a complex of 146 base pairs of DNA wound in 2 turns around the outside of a disc-like complex of 8 proteins called histones. A chromosome is a single piece of coiled DNA containing many genes, regulatory elements and other nucleotide sequences. Chromosomes also contain DNA bound proteins which serve to package the DNA and control its functions. Here are a few questions for you to work out. Differentiate between eukaryotic and prokaryotic chromosomes. Elaborate on the fine structure of a chromosome. What are histone proteins? Explain with suitable examples. Which are the different types of chromosomes? based on the position of centromere. Here are a few books for your reference. The Cell, A Molecular Approach, written by Jeffrey M. Cooper, Robert E. Hausman, 3rd edition, ASM Press. Biology, written by Peter H. Raven, George B. Johnson, 5th edition, published by McGraw-Hill. Advanced Molecular Biology, written by R. M. Twyman, 3rd edition, published by Bioscientific Publishers. Thank you for watching this program. See you next time. Until then, bye.